Good evening. My name is Carol Christ, and I'm the Chancellor at UC Berkeley, and I have the great pleasure of welcoming you to tonight's event. Before we begin, I'd like to thank David Raulet, who's sitting right there, Julia Schaletzky, and Mike Botchin for all of their work in setting up this evening's screening and panel discussion. I'd also like to thank Uncommon Productions for giving us permission to show their documentary, Jim Allison, Breakthrough, which will be publicly released this fall, and I understand there'll be a showing actually here once it's publicly released in the fall. And most of all, I'd like to thank Jim himself for returning to our campus to be with us tonight, for continuing to be a wonderful ambassador for Berkeley, even now from his perch in Texas, and for being the extraordinarily brilliant, driven, creative, empathetic leader that he is. It's an honor to celebrate him and his life-changing work this evening. Part of the documentary that you'll see in a few minutes features a young woman named Sharon Belvin, who was a graduate student at West Virginia University when she was diagnosed with metastatic melanoma in 2004. At the time, advanced melanoma was a death sentence. When it is localized, it is threatening but curable, but when the disease spreads to other organs, it becomes a relentless killer, resistant to radiation, chemotherapy, and until recently, to everything that doctors could come up with. By the time Sharon's cancer was discovered, it had spread throughout her chest and liver, and a large tumor was collapsing her left lung. After one of the new treatments then approved by the FDA only succeeded in sending her to the ICU, she wasn't sure whether she should continue fighting or enter hospice care. Sharon's doctor, however, proposed a last option, a new drug that would come to be known as ipilimumab, or IPI, which was then beginning its phase 1 trials. IPI was based on a new approach to fighting cancer, the first in a class of drugs called checkpoint inhibitors that did not attack cancer cells directly, but instead empowered the body's own T cells to fight malignancies. Sharon received just 4 treatments, an infusion and several injections in her leg, and 6 months later, her tumors were completely gone. Not only had she fully recovered from the brink of death, but her body's own immune system, not a surgeon's scalpel or high-energy X-rays or toxic chemicals, had been the one to save her. IPI's phase 1 trial represented the start of a seismic shift in the cancer treatment landscape, turning the field of immunotherapy from a long-shot intervention into a pillar of oncology. This shift, it is generally acknowledged, was largely built on the work of a single person. It's rare that such a sea change can be traced to any individual, wrote the science journal Cell in 2015, but the advent of checkpoint therapy would have been highly unlikely without the efforts of Jim Allison. Jim has devoted his life and his distinguished career to helping modern science better understand T cells and take advantage of them for treating disease. In his early days at Scripps Clinic and Research Foundation in San Diego and at the MD Anderson Center in Smithville, Texas, he committed himself to understanding the means by which a T cell recognizes an invader in the body. Then, once he joined the UC Berkeley faculty in 1985, he teased out how that new knowledge could be used to create a drug to combat cancer. And finally, over the course of an arduous but ultimately successful decade-and-a-half-long quest, he fought for the development of such a drug, convincing skeptical scientists and risk-averse drug companies that immunotherapy could not only work, but save thousands of lives. Last year, he won the Nobel Prize in Physiology or Medicine for his life's work, though, as he says himself, there's still so much more to be done. While Jim is no longer at Berkeley, but now the chair of immunology at the MD Anderson Cancer Center at the University of Texas, I know he retains a great fondness for our campus, his time here, and what Berkeley encouraged and enabled him to do. Furthermore, he's a wonderful example of what we preach, that our institution is no tucked away ivory tower, that there's a great benefit into bringing together the world's brightest minds to examine the pressing problems of the day, that we should cast a critical eye on accepted wisdom and the status quo, and that we must translate basic science discoveries into practical applications that affect humanity in profound ways. Jim, you may be a longhorn, but we will always proudly count you as a golden bear. Thank you, and thanks to David, Julia, and Mike for organizing the special screening of Jim Allison Breakthrough. It's astonishing how few documentaries there are about gifted scientists, and it's a joy to see this monumental discovery shared so widely, and to see a scientist like Jim recognized for the hero that he is. And with that, on with the show.
Hi. Wow. You know, that's the second time I've seen the film. The first time was on a plane on my little computer, and I have to tell you, it was a lot better in the big screen. And I hope that it wins an Academy Award. I'm here to actually introduce the panel discussion, and Julia Schaletzky, who together with Dave Raulet were the folks who brought this idea to have this filming here. I have to thank Jim and Pam for their generous spirit to come here and actually participate. This is a special trip for Jim. I have a few things I'm moved to tell you. First of all, I met Jim 35 years ago when I was on the search committee with Bob Tejan and Steve Martin. Steve is here, and I can tell you that when that masked man from Texas came, we didn't know what was going to hit us. It was actually, and this is very important for me to say, because it was really Bunny Koshland who actually told the three of us who were supposedly picking the candidate that this was the person that she wanted to recruit to Berkeley. She was the founder of the Department of Immunology. And Jim came, and of course the discovery of the T-cell receptor at the time, I didn't appreciate how marvelous a discovery that was actually going to be. I mean, the immune system, as many of you know, is sort of like a brain. It's a mini brain. It knows who you are. Your immune system can recognize you from everybody else. And Jim had this inspiration that he was going to cure cancer, and by God, he did. And you know, it's just inspirational to me that 35 years later, he's still taking it further to the next step. Okay, so I want to introduce Julia, who's going to come up here and lead the panel discussion, and Jim is, and the folks who are in the panel, some of them are the stars in the movie, and there's one announcement that I want to make, and I've asked Jim for permission. Turns out that his birthday, I think, is tomorrow. Is that correct, Jim? Yeah, and the co-star, the supporting star of the film, Lou Lanier. Is Lou here? Well, anyway, he's not here. Anyway, he's going to take Jim out sailing tomorrow, which they do, and maybe they'll make it to the Farallons. Hope you make it back safely. Okay, so anyway, congratulations, Jim, on your birthday. All right, so I'm going to introduce Julia, who will introduce the panel. Julia came to Berkeley 2 years ago, and it was really an amazing stroke of luck that we actually had a position to recruit her, because she's a woman of tremendous talents. Julia actually is a scientist. She got her PhD in Germany, working in Germany. And then she came to the United States to work with Tom Rappaport, who's a good friend of many of us here. at Berkeley. And Julia could have gotten an academic position if she wanted to, probably. She started to work in the Bay Area at Cytokinetics, which was a company that she and her team actually developed drugs that actually went to the market, phase 3, first of a kind. Not market yet, but yeah. All right. And we were able to recruit her here to Berkeley as the executive director of the Immunotherapy and Vaccine Research Institute, which is coupled with our first drug center here at Berkeley. We actually have a drug center that many of us here feel that we're going to be, although not a medical school, we're going to succeed in developing drugs. And the immunology division now in this mega department of MCB that Jim Allison actually built when he was here is now greater than ever. And we're going to hear some panel discussion of what it was like for Jim when he was here. And I'll just give the floor to Julia. Thank you. And we have some very heavy hors d'oeuvres for those who after, and I hope that you have the patience to stay because the food's good, and I hope we can just get together and talk about how great the movie is. Okay, thank you.
So thank you all for coming. It's my pleasure to introduce our panel today. I think most people will seem familiar after watching the movie. We have Alan Korman, Rachel Humphrey, Jim Allison, Pad Sharma, and David Raulet, the faculty director of IVRI here at UC Berkeley. And I've been watching the movie several times, and every time I just have to say, wow, what a story on so many different levels. I think is amazing. It's wonderful to look at it now in hindsight, what was achieved. It's also amazing how you pushed it through. So the first thing I wanted to talk about is how did you keep going? I mean, I look at the pictures of your family, your son is growing, he's growing. Jim is still persevering. How did you make it happen?
Well, I think maybe some of it has to do with what my brother noticed when I was very young, diamond head, meaning hard, I don't know. I think I felt, as I said in the movie, that we'd found something that seemed to me was going to work. And I was aware of all the failures in immunotherapy. I was also aware of the difficulty it is in developing drugs for reasons that have sometimes nothing to do with science or anything, but have to do with legal issues. I mean, the whole thing that bugged me for a long time was that people said, well, BMS has a patent on their antibodies, so you can't do anything, And I'd say, well, but it's bullshit, their patent is backwards, They got the biology exactly backwards, and to shut down possibility of a whole field because of that is ridiculous, And so that made me mad. And anyway, but most of it was just, after we cured in mice, I mean, it was like nothing anybody had ever seen at the time. We cured essentially anything. We went after either with a single drug or in combination. And I said, I can't, it deserves a test, at least. It deserves to get into clinical trials. And for it not to get there was just not acceptable.
Right. But you were fighting for a long time even before that, right? You saw something there in the academic field.
That was only 15 years.
Right? The mouse data was very strong, but you kept going, even with the risk, maybe in humans it's totally different, right? Who would have known, right? Yeah. That's where Pam came in.
But to not test it, to not give it a test was what, I was pretty sure it'd work, but maybe it wouldn't, but to not test it, to me, it was just not acceptable.
So you've been pushing very hard on this, obviously, but you've also been able to recruit around you people who also believed in the same thing and were also pushing very hard. I mean, I'm looking at Pam, I'm looking at you, looking at Alan, everybody had to pull their weight to really make it happen, and that's the thing that moved me about this movie is it shows beautifully how, university research, basic research has to dovetail with medical research, patient consideration, biotech industry engine to bring it to the market, and also with government and regulatory authorities, right? And in a way, it's also, can you inspire people? Do you find believers who believe in the same thing and push hard for it? Right. So how did you do that?
Well, I don't know. In my lab, I think I developed a certain rapport with people. I mean, Max Krummel talked about it in there, that people that sort of catch the bug. You don't have to think like everybody else, think differently, do what makes sense and work on it. But, and at some point, the data, I think, spoke for itself, with Alan getting involved and Rachel later on and Pam. It's the you look at it, and I think, objective person has to look at this and saying, wow, this needs a test. This needs to get forward. And as Neil said, when we first got together discussing actually moving it, the thing was, I just kept saying, you got to get it into people fast because we've wasted so much time over the last few years. And boy, they set a record. That was true.
It's true. It was amazing fast. Alan, can you talk a bit about the moment at Medarex when you decided you were going to do this? Because the risk was huge at that point, right?
Well, it actually started in... Is that working?
Yes. Yeah.
Well, it actually started at Nexstar Pharmaceuticals in Boulder, 1996. So... project started in 1996 in Boulder, Colorado, and then got transferred to Medarex. I eventually joined Medarex, and then BMS, bought into the program. In fact, I remember a meeting where Jim and I went to Bayer in Berkeley to pitch the drug. And a number of years later, I had a piece of paper that had Rachel Humphrey at the meeting, but she wasn't there. She was on the phone listening in. But that was way before BMS bought into the drug. So I hope you got a sense, though, of how long it actually takes to make a drug and get approval. So
after the super fast first time in human study.
right. So
Well, I think the only way they actually did it that fast was because they started before we actually signed the paperwork.
Right.
first patient was treated in 2000, but the approval, we actually saw the data in 2009, and that approval was 2011. Mm-hmm. And
March
as I like
31st.
to say, we still don't know how anti-CTLA-4 works in 2019.
Right. Tell us more, Rachel. You had some.
Yeah, I just want to reinforce what Alan said, actually.
Mm-hmm.
And I think I said it in the movie. I was at Bayer listening to the pitch. Mm-hmm. And I went to Bayer and said, I think we need to get this. I remember trying so hard. I went to Germany. Pay attention to this. I'm telling you, pay attention to this. And they wouldn't. They said BMS has the IP, the intellectual property. So I moved. Mm-hmm. And I went to BMS, and I came in on the day of the due diligence. Mm-hmm. And I think that's what you were saying before, which is we built a team at BMS 15 months before the deal was signed with Medarex, and headed to the FDA with members of the team with no deal, just to be part of it. So there was, and then you and I connected every month in New York. We were determined, all of us, to make this happen under any circumstance. We were a team. So.
That's amazing. I think also interesting is that, I mean, in hindsight, you guys all saw it, right? Everything worked out, but there were so many people who were like, I don't believe it, right? I see the data, but I don't believe it. Right? Yeah.
Let me add one thing, and then you may want to say something more. I think the movie did a nice job of introducing the following concept, but I'm not sure folks fully appreciate it. If the tumor grows first, before it gets smaller, it means clinicians have to be willing to hold off switching to something else before they, to give the drug a chance to work. So it's actually very important that folks like Pam and all the doctors that were working on the project with me, I'm a clinician too, could feel okay about it. And we have lots of examples of individuals who refused additional therapy, came back with their tumor gone. So it was a revolutionary time where the people who were pushing back was not just at the scientific level, but even at the ground level with patients and doctors. And now the standard of care is to wait. This could still be working. Everybody knows. You just don't push forward. But imagine 15 years ago, if someone you loved looked like the tumor was growing, you want to get them on something else fast. It's not simple.
It had certain things working against it that I realized at the very start. I mean, we were treating cancer by ignoring the cancer cell. We were not treating the cancer cell at all. We were treating the immune system, which meant all that other, the fact that it worked late and took a while followed from all of that. And so it was so unconventional. As Rachel said, people were afraid of it. And it was judged, one reason that Pfizer's trial failed is they treated it like a cytotoxic, like it was actually going to kill the tumor cells. And so obviously if something grows in the face, if something's supposed to kill them, it failed. But that's not the way this drug works. And I had to keep telling people, follow the biology. But as Rachel said, that's not so hard when you got to say, my patient's getting worse, but I got to hold off.
Right.
Do we know why it grows first? Before?
Cells can infiltrate into the tumors, and I think that's what we found. I think the other thing I want to say to all the students is that the reason I got to...
Closer.
Oh, so is that better?
Yes.
Okay. I think, for all the students out there, the reason I got the chance to work on this is because it was such a brand new concept that when Medarex was trying to write the trial to get the oncologists and the more senior oncologists involved, they didn't want to be involved. They were saying no, they're not interested because they have a lot of other programs they're running. And so I was in Lloyd Old's lab at the time. I did my PhD in immunology and my postdoctoral work in Lloyd Old's lab, but I was a fellow at Memorial Sloan Kettering. And so they came to speak with Lloyd Old because he was the father of tumor immunology. He discovered CD8 T cells, and I was there at the time, and I was able to be a part of that conversation. And so because I was so interested and I wanted to do it, and they allowed me to have a say because they couldn't get more senior people to work in it. And as a student and as a fellow, I was able to jump on it and say I wanted to work in it, and that's where it takes off. So I think for the students, you're so new to the field, and you're willing to do a lot of different things, and you don't have tunnel vision, as Max puts it. You're able to see things broader, and that's to your advantage. So I always say to students, don't forget to take advantage of the fact that you can see many, many other things that more senior people may not be able to appreciate.
Yeah. Out of the box thinking, very important. I think the movie shows that beautifully too. So, if you think back to your time at Berkeley, you always speak fondly of being here. I think for me it's a place that really fosters innovative, out of the box thinking. So, what did Berkeley get right about the process? Because it needs a special place to also give that opportunity, right?
Yeah, well. When I was at Smithfield, one of the things that was in the movie that I loved about it was there's nobody at all around that knew anything, so I could do whatever I wanted to do, and everybody thought it was cool, because there was nobody. Right. But when I came to Berkeley, with a little afraid of coming here, what I learned was so many excellent students, and it's easy to get postdoctoral fellows came to it just because it's got a reputation for a center of knowledge and advances and things in the faculty, by and large, are very supportive of that kind of thing and look forward to pushing the envelope as well. And I think in the immunology group that we had, it was a very collegial group, and we always challenged each other, and the students. It was just this atmosphere. If you put something out, you better be able to defend it, not in a mean way, but to say, argue it. At least the training my students got, and I think it was true in many of the labs, was somebody says something, your job is to find a different explanation. for what they said, and that level of skepticism and open-mindedness, just keeps things churning until you finally get it right.
Mm-hmm. Mm. Yeah. David, you want to elaborate a bit on the environment in Berkeley?
Yeah, I mean, I think, Jim helped build our group, and he, basically we've used his work and the current immunotherapy work as inspiration to develop new approaches for cancer immunotherapy. That's what we're working on now. And I think as someone in the film said, which I thought was a good point, that these are the early chapters, and they're amazing chapters, but there's a lot of chapters to go. A lot of cancers can't be cured with the existing immunotherapies or any therapies, and we all need to work hard to find new ways to do that. And so that's why we formed the Immunotherapeutics and Vaccine Research Initiative at Berkeley, to try and find new ways to do that kind of work. And so what we do is we bring together people who work on cancer immunotherapy and people who work on infectious disease immunology, and we have them talk to each other in ways that doesn't occur everywhere, and we try and come up with new basic science approaches that we can try and translate into new therapies. And it's really, I think, due in part to Jim's inspiration that really led us to realize that this kind of thing can be done. It's been done before. There's a long way to go, but we have new ideas, and we try and come up. I mean, if I can just give one example, T cells are great. They can kill a lot of tumors, but it turns out that they may not be able to kill all tumors, because some tumors are maybe invisible to T cells, like they don't have any antigens or presenting molecules that T cells can see. So we try and come up with ways to target other immune cells to mobilize them against cancer. I work on natural killer cells, and that's really another kind of immune cell that has a different mode of recognition than T cells. So they can recognize a lot of tumor cells that T cells can't, but they can also kill tumor cells just like T cells can. So we're finding ways to activate them and prevent them from being inactivated to try and mobilize responses. We have really nice data in animal models, and we're looking to translate that work into new therapies for patients. And there's really a lot of other work going on along those lines, and we try and foster cooperation with industry partners who are also helping to fund the research we do, and that's been very productive so far. Yeah.
Thank you. Because for me, the interesting thing here was also, how do you foster an environment where you can think out of the box and come up with something completely new? And in reality, if you start your career, you're always dependent on buy-in from others, right? You have to have support from colleagues. Your papers have to be favorably reviewed. If you do things totally against the grain, it's very, very hard, right? So I think also currently there's a lot of new research happening. Some might be good, some might be bad. How can you tell early on? It's very hard. And how can we enable people who do good science but think out of the box to actually use that instead of just going with the flow, right? And so I think philanthropy plays an important role there too, because how do you fund these early things, right? Normally government funding is very much for things that are already pretty established, and then you just keep going, everybody buys into it, right? But what about these really new findings? How can we support research going on there?
Well, it's a real issue, I think, because one of the things, the NCI and IH are definitely the engines that drive the vast majority of work, but for most investigators, it runs on a 3-year cycle, and so it's difficult to take the long view there, and so it leads to not making any waves, extending stuff a little bit, and I don't know any better system right now, except there needs to be a multitude of systems. I mean, as you said, philanthropy is another one that can work. Things like Howard Hughes Medical Institute. I mean, I think that the more different ways of funding science that we have, the better off we are all across the board. Because we need that steady progress, but we also need to have the ability to take the longer view.
Right.
And so.
And also take the risk, right?
Yeah.
Knowing that the majority of things will fail, but without that, you can move the things forward that succeed, right?
The first patient to get anti-CTLA-4 actually was a prostate cancer patient, not melanoma. And that was done because the Prostate Cancer Foundation was very involved. So when we wrote the trial, we were trying to, because of Jim's brother's story and the Prostate Cancer Foundation providing the philanthropy, that's how we actually started that part of the study. So philanthropy is very important for getting the first, set of data that then I think brings other people on board.
Because I think particularly the industry part of that story, I mean, it's clearly once there's success and somebody has done it, everybody wants to get in, right? But before, to do that first round is the hard thing.
Well, there certainly is a frenzy of activity now. So there are a lot of biotechnology companies who are investing in this area, and there's a lot of venture capital that's entering that. And so it's a particularly good time for that, but we know that that's a cyclical phenomenon, and hopefully
A few years ago
lasts
at
a bit
meetings,
longer.
I'd finish talking and students would come up, and our colleagues and the people come up and ask questions, could you extend on this? I've got something and whatever. And then, I don't know, about 8 years ago, I think, it'd be venture capitalists would come up, and students, other people couldn't even get in. I'll give you $500 if you'll talk to me for 30 minutes.
Yeah, they're the only people that go to posters now.
It's probably worth noting, though, that 15 years ago, the Society for the Immunotherapy of Cancer, SITC, was there 50 of us? I mean, honestly, and
wasn't even called SITC.
Yeah, it was called
ISBTC.
It was called something else. I'm looking at you. I can't remember.
I remember when there were about 10 or 15.
Yeah, 10 or 15 people. And at ASCO, the annual Society for Clinical Oncology meeting in Chicago, it's in Chicago now, immunotherapy was such a poor stepchild that they put it in the furthest place in the building where walking in heels was a real issue. You had to actually take it off and walk barefoot. You got there and you stayed there all day because going back and forth made no sense. And it was usually on Tuesday when everybody was gone. And when, let's not underestimate, when Alan said, let me try it, these are centerpieces of the universe here. Absolutely no one was doing it. Absolutely no one. So everybody's jumped on now, but back in the day, it was a very lonely place.
Right. I think, not only was the cancer immunotherapy part hard, but I mean, even using antibodies as drugs was people were hugely skeptical of that. Now it's everything like that. And you have to ask yourself why, in retrospect, people thought antibodies wouldn't be good drugs. It really doesn't make any sense. And that's the kind of skepticism that these guys brought to bear on it that really made this happen, I think.
I think the other big thing that was revolutionary, and maybe the movie doesn't capture that well, is that oncology drugs for many years were developed in mouse models with no immune system. That's how most oncology drugs had been developed, because we did not even think of the immune system as playing an important role in cancer patients. Our models didn't have an immune system. And so for the first time, because of now the success of what an immunotherapy can do in patients, we have to rethink how we do the science in the lab to really make sure that there's actually an immune system that we're taking into consideration. And maybe that's why a lot of drugs in oncology failed, because we did not take the immune system into consideration. So even though we can cure the mice, we were not able to translate that to the clinic. So I think this whole concept changed so much, not just in terms of this particular creating a new field of immune checkpoint therapy, but just changed our thinking for so much of oncology, so much of immunology, and so much of translational research and patient care, to be honest.
The only thing I'll say is the answer to the question, I think, see if you agree, why antibodies, people didn't think it was going to work is because it was completely new. And then immunotherapies weren't going to work. And then cellular therapies, who could ever think about putting cells into somebody? And then engineered cells into somebody. And so the next question you have to ask, especially students, but all of us, any of us, it doesn't matter how old we are, is to ask the question, what completely crazy idea do you have that everybody thinks is a bad idea is probably the smartest one?
Yeah. Asking the audience for their ideas now. 10 years from now, Yeah, no, it's very true. But also realistically, the difficulty is to gauge, right? Because, everybody has a lot of ideas. Probably most of them aren't good ideas, quite frankly, right? And you have to pick the ones where it's really worth investing 20 years, right?
Ideas without data
Right.
is not that valuable, though. So.
Right. Right.
And ideas without pit bulls don't go anywhere.
That's true, too.
So you've all seen, I think, the process from innovation in the academic field through translation, eventually to the patient. So I'd like to ask all of you, if there was one thing you could change if you had the power, how would you make the process better?
I would have wanted it to be faster. I mean, 15 years is a long time, and we lost a lot of patients. And as you heard, Jim's brother was somebody we really wanted to treat. We were trying to write the trial so that Jim's brother could be a part of that treatment. He got very sick and couldn't be enrolled. So there are patients who died along the way 15 years. So making it faster is always, what I wish we could do different. But there's regulatory process and, the funding process, and there's just all these different steps that we really have to take into consideration as well. But getting it to patients faster would be nice.
Be good. So which could be achieved through, infusion of more money or more, easier process in general to enroll patients? Or where do you think is the biggest obstacle in terms of timing?
I think sometimes we're a little bit hesitant to write that first trial. I know that's what took Medarex a while to really get people, like Rachel said, it took a year to enroll. Even now when we write clinical trials, they don't enroll. 15 to 20% of patients maybe will enroll, that's it. So we still have a population that don't enroll in clinical trials. We're not very good at explaining it. We use a lot of scientific jargon. We're not Scientists are not the best communicators to get their ideas out. We have to do a better job of that, I think. And I also think we need to see that clinical trials can be written for 10 to 20 patients. Maybe we don't have to write an 800-patient trial to get our ideas understood better and get some data to help build from that. So these are things we're learning now.
Yeah, getting away from the sacred p-value would be a good thing, because now with the advent of these IO agents, if you don't see a responder in 25 patients or so, people move on.
Right.
It'd be useful to get some tissue from the patients and see what happened. So you'd have a little science behind it. But if you have to do 800 patients and look for a statistical difference between the treated and untreated, is it worth it? Because that over the decades, I'm convinced, just led to more of the same. And so people they weren't really looking for big advances. 3 months is fine. If you could get a p-value for it.
Yeah, I would say, to answer your question, Julia, we should spend more money on research and less money on walls and bombs.
Yeah, I think we all agree on that.
And I should just back up, it's just not evident, all of the whole story that you saw the movie about tonight came about from knowledge of basic fundamental knowledge on how T cells work. It didn't have really, the cancer part was in the back of the mind. It was not the reason the work was done, and was not done for the purpose of trying to say, let's get a new cancer drug.
I have a question. How do you remain so honest? It is so wonderful to have scientists like you who have won the Nobel Prize. I know so many who are not 1/10 as good as you are, or they should have never gotten it, and they are so arrogant. Oh my God. So, how do you do it, Jim?
I don't know. That's just.
I think it's Pam.
I'm still working on my harmonica. I'm not nearly as good as Mickey Raphael.
So Mickey told Jim he has to stay a scientist and not try to get to be so good on the harmonica. But I think for Jim, I don't know how many people have seen Jim around a patient. When he meets the patients, it's always the most moving thing. And clearly, I have clinic every week, so we meet a lot of patients, and he knows a lot of the patients. There are ones we don't cure. They're ones who participate in the clinical trials, and they do die from their disease. And yet they still come to thank us and thank Jim for, letting them participate in a trial that, doesn't maybe work for them, but he wants to always cure everyone. And I think that's something to keep in mind. They ground you. The patients ground you. They make you realize we have a lot more work to do. And if you've seen Jim around a patient, you'll know that's what keeps him humble.
All right. I think you wanted to respond, Rachel.
Yeah,
You had some ideas.
actually,
Going back
it was
from,
such a great point. I don't. The only thing I was going to say is you asked what could we have done differently. I think it's just worth celebrating for 1 second what went right.
Yes, right.
Because this is and you and I were talking about it actually when we were sitting over there. This is a unique blend that's to some extent a bit lucky, but it's just worth highlighting. Not just the folks around the table who, perniciously, doggedly, gave their career. But even at BMS, you saw a little bit about Elliot Siegel. Right. This is an extremely wonderful man who said, this looks interesting, go. Helping all the time, listening carefully, ear to the ground, super supportive. It's very rare to have just the right mixture of people. And so all I would say is that in your own work, find those partners who are willing to stick the plant in the ground with you, willing to take the risk, even personal risks, to do what they need to do to get your great idea going. I think that's the fundamental chemistry here that I personally feel very blessed to be part of.
Yeah, I have to say that was Lloyd Old's way of, Lloyd Old, I was in his lab at the time when this was all going on, and he was determined to make sure that we were going to do this. He believed wholeheartedly that this was the way forward, and asked me to make sure that I worked on it as the clinician, but also as the postdoc in his lab who knew immunology. And I think when he met with Jim, he told Jim, just keep sticking with it. This was going to work. It's these kinds of people who make you say, no matter how many naysayers there are, you need the one person in your corner. So it's important to have that.
Right. I also meant in terms of policy in general, what could be different? Like if you could just do executive fiat and make the process better,
Get rid of all the lawyers every time
exactly,
we have to.
Right?
Every time I have to do a clinical trial agreement, we have more and more lawyers.
That is true.
That's right, get rid of the lawyer.
Right. And also every hospital a different agreement, right? That
yes,
always
yes.
shocked me. Like
Let's not forget the tech transfer offices
The tech
at universities.
Intellectual property.
Intellectual. So yeah, maybe at this point we should open up. Yeah, please. Did you want to respond first, or should we open up to Q&A? Okay, maybe it's time then to go into the Q&A. You've started. A little bit. Yes, a little bit. Okay, let's do 3 questions. No, there's no mic. Just speak up. We could hear you. Right.
Thank you very much. She wouldn't let me talk. So, you once said to me that you wanted to come back to Berkeley and just sit in your boat and go around and around. Why can't we have you back?
There's still a lot to do, We're only up there with about 60% response rate in melanoma and 40% in kidney and lower in prostate and nothing in glioblastoma or pancreatic, and so there's a lot of work still to do.
But we can have you do it here.
Hallelujah.
Maybe.
Okay,
Maybe.
some more questions? The back.
Jim, I've always been puzzled. Weren't you a little bit concerned that by releasing the brakes, you just have a storm of autoimmunity released? Did that concern you?
Well, yes, it did until we did the experiment and nothing bad happened. I mean, we never saw any adverse events at all in mice, other than a little vitiligo when we were curing mice of melanoma, which was to be expected. In the cynomolgus monkeys where the safety tests were done, there were no problems at all.
But
But when it went into people, there were, and perhaps Pam can.
We wrote the trial with a safety and stopping rule, and we wrote it at a 3+3 design initially. So the doses were changed, we've started very, very low and then increased, and there were stopping rules for if 1 out of 3 had a certain amount of toxicity. So it was written very, very strictly, required by the IRB at the time that way. So that cartoon is actually wrong in the movie. And we've said that to Bill Haney many times, and he says, Pam, I can't put an antigen-presenting cell and all of this in between. It makes it too complicated. We'll keep it simple. So it's not quite the tumor cell. It's more of an intrinsic property of the T cell.
The CTLA-4 works. It's hardwired into the system. You have CTLA-4 to stop T cells from killing you, basically. And so once we figured that out, I thought, well, if you release it, that may be bad, On the other hand, patients get a lot of itises, pneumonitis, colitis, things like that, but they're really not autoimmune, and that the patients can be treated with steroids and then weaned off of them, and they don't come back. That said, there are patients now that so many people have been treated that do develop things like rheumatoid arthritis or type 1 diabetes. A few with a heart problem is poorly understood. So there is a price to be paid. The cost is not zero, but it's more, I would argue, it's a lot more acceptable than a lot of chemotherapy agents.
That's right.
So, Jim, just to follow up on Tom Klein's question, I've heard you say many times and taught me that it's really the cancer cell itself is producing all kinds of mutations and producing new proteins that are recognized as foreign on the cell surface. And that's the deep insight. And in fact, you've taught us that, in fact, those tumors that are have the most mutagenic phenotype are the ones that are most susceptible to the checkpoint inhibitors. Is that's an important point to emphasize here.
Yeah, most of when we do checkpoint blockade, our PD-1, it's releasing T cells to see mutant proteins, and so it spares most normal tissues, not always. But what happens, I think, when patients develop colitis and stuff like that, is just an inflammatory response gone amok due to too many enchiladas for dinner or something, that cause some irritation, and it's not controlled. But
I'm ready for an enchilada myself.
Yeah, exactly. We got the message. So thank you so much. What? 3 questions. Okay, one more. Last one.
I just wanted to make a comment about the film as a total layperson, non-scientist. Everything seemed so clear, and I never glazed over as I usually do, and it was very inspiring and educational for me.
That's great.
A lot of credit to Bill Haney and Jennifer Pierce and Maura McCarthy. They did an incredible job. They followed us around for, I would say, over a year. We did not see the movie. We had no idea until it premiered at South by Southwest with a big audience just like this. So they did that all. So all that credit goes to them for doing a great job with the movie.
Right.
Yeah, can I just say, I think we really should thank Bill Haney and Jennifer Pierce for allowing this movie to be shown before the theatrical release. That was really great of them.
Yes.
We really appreciate that.
Yes.